So up next, it's going to be me and Jairam and I talking about NeMo and EpiViz. So you can see the screen, correct?
Yep, we see you.
Yep, looks good.
Yep. So Mike mentioned a little bit about the NeMO archive, NeMOAnalytics, and how they're kind of two different ways to approach making data easier to view for things. So in this talk, we talk about analytics and EpiViz, which is kind of integrated with NeMOAnalytics for looking at particular types of data. And so NeMO archive is where you can think of, that's where the data may live in its raw format or in any of those different level formats that Mike was mentioning previously. On the NeMOAnalytics side, this is where you have a number of different tools for data visualization for either gene expression or epigenetic data, comparing between different data sets, and then some tools for statistical comparisons as well. So the archive is kind of where the data is housed, and then the analytics platform is a online platform to visualize and look at data. And our goal of having the NeMOAnalytics platform is to support BICCN generated multi-omic publications. So to make the data available in a easy to interpret and query format, where maybe if you have a particular question about a gene and you want to go and look at the data from one of the marker papers or one of the other flagship papers, but you don't necessarily need to do a deep dive into the particulars of the raw data itself. So the NeMOAnalytics is built around a kind of a gene-focused approach towards data. So within data sets, looking at things from a gene point of view. And one of the things that you may see in some of the publications is, let me see, is if you end up going to the Nature paper or Nature and looking at some of these captions, you may see something that looks, it's like, here's an example from one of the papers. And so you have your figure, and I think Mike showed this figure earlier as well. But one of the things you can do is you can explore that data in analytics just by clicking on the link, and then it'll bring you to that information there. In NeMOAnalytics, you have tools to look for a comparison between bulk RNA-seq data, so looking at gene expression between different groups. We have a single cell workbench, which pretty faithfully replicates a Seurat pipeline, so going from filtered count data through visualization and analysis, doing cell clustering and along those lines. We also have the EpiViz, which is for viewing epigenetic data, and some other visualization tools like the multi-gene viewer, which is where you can create heat maps, volcano plots, and other things where you may be interested in looking at a comparison of more than one gene across different groups. And to get to NeMOAnalytics, it's just NeMOAnalytics.org here. And you'll end up at a homepage that looks similar to this. When you first log in, you can create a profile in the top corner here. The profiles are free to make. You can run them yourselves, but but it allows you to kind of save your analysis as you go, to upload data sets and things, and some other functionality as well. So I'd recommend, if you're going to play around on NeMOAnalytics, it helps to make yourself a profile. And there are also things, so you have your user profile, which is kind of your intro, in this case, on one of the curator accounts. But you also have profiles of data sets. And this is where groups of different data sets, or maybe a single study, but multiple different types of analysis, can all be kind of put together into one place. So on here, a lot of the highlighted profiles are related to the flagship paper and other things. So I'll go and take a look at our motor cortex cross species profile, and we'll just do we'll take a look for the GAD2 gene here. Once you hit enter, it'll take a second or two for the different profiles to load. But now you can see the visualization for a number of different data sets within this profile. So in this first one here, we're looking at the motor cortex in humans, where on the left you have the expression of that particular gene of interest. And on the right side here are the class labels of the different cell types. And you can see that same kind of format, where on the left is your particular gene of interest, and the right, the subclass labels, follows through for the different comparisons. So in this case, the second case here, you have the mouse, marmoset, and so on for whatever graphs are in that particular profile. Up at the top here is where the cross-species part can come into play. So in different species, you may have a similar gene, but it may be named slightly differently. So in the case of the mouse here, so this particular gene is GAD2, but if in humans, that one doesn't necessarily exist exactly. It's a slightly different name for that gene. So you can click through the different organisms and see what that annotation matches up to for the different groups. So in mouse versus humans, and there are other ones in here as well. So zebrafish, chicken, rat, and marmoset. Also, when you click through, you can find links to learning more about particular genes. So if you want to link out to maybe the ensemble IDs for that particular gene in that organism, or other items, you can easily do that from the link there. And you can do the same by, if you wanted to look at different genes of interest, by just typing them into the box. And then they'll show up in that gene in the profile. So this is really nice if maybe you went and you looked at the flagship paper or some other papers, but they never went and really went too far deep into your particular gene of interest. So this is an easy way to go and look through other genes that may be of greater interest to you than others. So you can do that in here, and there are a whole variety of different profiles that were made by authors or our curation team, looking at either the different particular papers or combined profiles where we may have put together data sets from multiple of these papers. You can also search through our dataset explorer if you're interested in a particular data set. So, and this can be subset by organism, data set type. So if you're interested in epigenetic data, for example, you can subset by that, or you can enter some search terms in the top to pick out data sets of interest. And when you're in the profiles, these also offer an easy way of downloading this data as well. So on this little dropdown are a number of different extra things you can look at. You can use the view tool to zoom in on a particular one. Info just provides some general information. If that particular publication has a PubMed link, that will link out to it there. And it'll also link you to things like Geo or some of the various analysis tools. And if you want to download this particular data set right from the NeMo archive for public data, you can just use the download options here. So there are also, so these graphs were graphs that were made on using NeMOAnalytics, but there are also a number of kind of specialized graphs that were made for, in this case, we'll talk about some of the ones for the BICCN. And you may have saw them in Mike's talk, but for example, they offer visualizations where we're integrating, in this case, the patch-seq data and electrophysiology, but now you can add in your gene of interest. So in this case, GAD2 is in here, and you can see the expression of that gene across the groups for whatever gene you are interested in. So you're not dependent on the figure from the authors or remaking a whole figure to see differences in these groups. So here's an example of doing it in patch-seq. There's also a similar visualization for working with Merfish data as well, where it's showing you across that that spatial profile as well. And at this point, we're going to talk about one of the tools, so related to epigenetic data. And so, Jairam, I'm going to pass it off to you for EpiViz.
All right, cool. Thanks, Joe. Can I share my screen? All right.
Yep.
Let me know when you see the screen because I can't really see it when I share.
Yep, it looks good. If you, or did you want it in presenter view or just normal?
Yep, well, let's go to that. Is it full screen?
Yep, looks great.
All right, thanks, Joe. So Joe talked a little bit about where the data is and how NeMOAnalytics allows you to visualize or explore these data sets. So NeMo Archive is the place where all of the data is hosted. You can also use Terra and reprocess the data and then store the results back there. And you use NeMOAnalytics to visualize it. And one of the functionality that we integrated as part of NeMOAnalytics is the ability to directly visualize any of the processed files that you have generated as part of the pipeline. For example, you might have generated cluster level, big files or pseudo bulk files, and you may want to explore these data sets or files for different epigenetic data sets or methylation data sets. So you can use EpiViz for this. I'll talk a little bit about EpiViz, and then I'll show you a couple of works profiles that contain epigenetic data. So EpiViz is very similar to other genome browsers that you might have used, especially the UCSD genome browser or HEV. It uses the linear organization of data and displays data sets as tracks. It also integrates a lot of annotation information, for example, information about genes or other features, for example, CPG islands, promoter regions, any annotation files you can you can visualize them in EpiViz. There's also ways to quickly navigate along the genome in EpiViz, but a majority of the genome navigation in NeMOAnalytics is controlled by the gene that you search in a profile, and it automatically is set. The view is set to that particular gene region. One of the interesting features or intuitive features is we provide a number of visualizations for interactive visualization of functional genomic data. So in addition to track-based visualizations that you normally see in genome browsers, we also support plot-based visualizations. And this includes, for example, heat maps, if you have gene expression data, or scatter plots, if you want to make something like MA plots. And for most of expression data sets, they're much more intuitive to visualize and explore them as heat maps rather than tracks. So EpiViz supports all these types of visualizations. We also have brushing implemented. This goes across plots and tracks as well. So when you mouse over a track, it immediately provides a visual cue of what the region is across all the other data sets on the EpiViz panel. And these workspaces are profiles that you create on NeMo are shareable so that you can use them in multiple profiles. So for the purpose of the BICCN, we have a couple of workspaces that we created. I don't know if you see my pointer. Let's see if I can get my pointer to work. Oh, there we go. All right. So for the purpose of the BICCN mini atlas, there was a flagship data set that was generated for 6 different types of major clusters, and there were several minor clusters in the data set. So we generated pseudo bulk files for all the epigenetic data in the peaks. For example, in this case, we're looking at ATAC peaks across glutaminergic and GABAergic cell types. We can also explore the signal for all of those major clusters that we see, and the methylation data displayed as tracks for a particular region of the genome. Similarly, EpiViz also has the ability to aggregate data on the fly. So if you don't want to look at individual cell types, you can also aggregate them to a higher label. For example, in this case, instead of looking at the minor types, maybe you wanna create an aggregated track of all the glutaminergic cell types as a single track rather than separate ones. So you can do that in EpiViz. You can specify what we call as a computed track, which is computed by using the mean of all the cell types that fall under glutaminergic. That's the red line, the red track that you see, and the red line is the GABAergic. And you can do similar things for both ATAC and methylation data. We'll do a quick view of one of the profiles. This one. So this one is the same data set that I was showing before. We're looking at the GAC2 gene region. So this comes from the publication itself, where they published the t-SNE coordinates for this data set. And you can see the GAC2 gene expression that is overlaid across the t-SNE plot. It shows you a lot of highly expressed in the orange region, like over here, and a lot of in the brown cell type regions over there. You can also, in parallel, when you're looking at expression data, if that data set also has methylation or any of the epigenetic data, you can use those 2 profiles together or panels together and create a single profile on NeMOAnalytics. And when you go to a gene region, the EpiViz panel automatically picks up that gene and initializes the browser to that gene region. So GAC2 is always going to be at the center of the genome track. And as you can see, when you mouse over, you can see that region is being highlighted across all the other tracks in case you have hundreds of tracks on a particular profile. In this case, we're only looking at 6 specific cell types that is coming from the lab. These are the accessibility profiles for the 6 cell types and the methylation profiles for the 6 cell types. A few features that we provide, most times when data comes in, they all have their own scales. Maybe for visual comparison, you want the y-axis to be fixed across all the tracks. For example, in this case, the first track has a maximum value of 58, the other one's 50, 60, and so on. There's this icon that you can press that would collapse all the tracks to use a single Y maximum, so that you can compare them relatively across all the ATAC-seq profiles or ATAC-seq cell types in this case. There's a few changes you can do. You can also specify what colors you want to use for different tracks. That's changeable. There's also a number of settings that you can set for each of the track in EpiViz. Depending on what type of track it is, every track has a very specific set of settings. Common settings are, you can specify a chart title and stuff like that. But also, if you have a very dense data set, you probably don't want to visualize every single base pair in the data set. So maybe you want to aggregate data for every X number of bins. So you would use the step parameter. For example, you can say 15, and that would aggregate the data for every 15 base pairs and visualize a single point rather than 15 different points. You can also change a number of attributes with respect to the visualization itself. If you want to choose a different smoothing parameter when you render the line across all the data points, we provide a number of smoothing functions to use. The default is always basis, so but if you don't like basis, you can change it. I'm not going to apply that. This is another example where it's a very dense data set. I think the step value by default is 15, so every 15 base pairs in the region is being aggregated a single data point. But if I remove it, let me change the interpolation to basis and apply, you would see a lot more data points and a lot more granular scale. And when you zoom in, you might see a lot of the highs and lows in the methylation. But maybe for the purpose of visualization, especially at the gene level, you may not want to see that much detail. So you can start to aggregate data to a higher level. And that step parameter is especially useful for very dense data sets. Going back to the slides. All right. So in the EpiViz panel itself, there's a number of functionality that we provide on NeMo. So you can also, there's an icon right next to the logo that would open the EpiViz, the genome browser itself in its own window, and you have the full functionality of a full genome browser, and you can add more data sets or bring in more data sets that are available through NeMo Archive and continue to explore them or visualize them. In the full mode, you also get the full navigation panel. So you can navigate left and right along the genome or zoom into the region or outside the region, or you can also type your own custom chromosome location. Now let me turn on the pointer again. That custom chromosome location is over here. We also provide a search, so you can either search for a gene of interest and directly navigate to that region. And this is only available on the full mode, not on the panel when you see inside the NeMOAnalytics browser, because navigation there is controlled by what gene you search. We also provide a number of different types of visualizations. I'll actually go through them in the next couple of slides. But we have genome annotation tracks that provide you the position of genes or exons or also transcripts. And for some of the genomes that we support or annotations that we have, you can use what we call as blocks track in our system. It's a visualization used for visualizing peaks across multiple samples, and you can overlay them on top of each other. And similarly, if we have signal data, this is ATAC or ChIP-seq or methylation, you can also overlay them on top of each other and look at and visually compare across samples. A few features. This is something I've talked about during our demo during the profile. If you want the same y-axis across all the samples or tracks, you can set that by choosing a fixed axis. If you don't want that, you can click on it again, and it would toggle between a fixed axis versus a not fixed axis. You can also apply transformations over tracks. For example, if you use a line track as one of the visualization, you can either calculate a log2 ratio, you can mirror the plots so they're on the opposite side of the scale, or you can calculate a mean and plot them as a single track rather than two separate tracks. These are things that I've shown before, where if you have a very dense data set, you may not want to plot every single data point in that data set because it may not be visually helpful. So you can use the step parameter to aggregate data for every X number of base pairs in the region window. And this is available on most of our visualizations. If you're using heat maps, there's a number of options that are available on heat maps on how you want to choose your clustering method, and also the clustering, the metric that you want to apply. Do you want to apply over rows, which are your samples, or over your columns, which are your gene regions, or you can do both. In this case, that's the large part of the GIF. And you can also hide and show the dendrograms that were created to give you an overview of which cells or which genes or which clusters, which samples or cell types cluster together. One of the things that we've recently added is the ability to hide tracks. So if you have, if you're looking at a particular data set and has, hundreds of cell types, you can hide non-relevant cell types in your visualization. You can also rename these instead of these auto-generated names that were created. That provides a better way of only looking at the data that you want to see. You can also change colors of any of the visualizations that you see within the EpiViz panel itself, and that's possible through the color palette icon. And finally, what I've shown today is, or through the slides, is one of the profiles that we pre-created. I'll show you some of the interfaces that we have so that you can upload your own epigenetic data and create your own set of panels and add them to profiles. So if you're on the NeMOAnalytics homepage, once you log in, once you log in, there is the upload dataset panel, and under that, there's an option to upload epigenetic data. If you're not familiar with NeMOArchive, portal.nemoarchive.org is the place where all the data is being hosted by the BICCN and the NeMO platform itself. So you can go here, look for data sets, for epigenetic data sets, especially for big files, PET files, or any of the supported file formats that you can use for visualization. And once you have a set of files, we provide a couple of options on how you can add your own epigenetic data to NeMO platform. If you have an existing track hub configuration, which is very commonly used with the UCSC genome browser, you can provide a link to the track hub and we will automatically import or register those data sets with the NeMO platform. Or if you have files that are either on your machine that you want to upload to NeMO, you can upload them through this interface, the second option. Every file, we also provide access control options. So you can choose if a data set is public or private. If it is private, only you can access those data sets and create visualizations, and no one else can actually see those anymore. Well, no one else can actually see them. If they're public, everyone else on the NeMO platform will be able to see those data sets and include those visualizations in their profiles. You can upload more than one data set at a time. I don't think there's any maximum so far from what I've tried. Yeah, you can upload as many data sets as you want. And once you're finally done, click on submit, and that would register your data sets, or register or upload your data sets to NeMO. Once you upload the data sets, we have a section for a panel designer, where you're now creating one of the panels that would go into a profile. So you can provide a little bit of information about what type of data set it is, provide some description of the files or the profile you're creating. You can also set access restrictions on the panel itself so that the panel is not publicly accessible. And you must provide a genome that you will be using for this data set. By default, it's human, mouse, but you can also change it to human. And we have a number of And this one, the panel itself will automatically update. Once you're here, you can click on Add Chart. I have a number of data sets that I've uploaded before, but I can choose a couple of them and then add them. Once the data is being accessed, the plot automatically shows up. You can change the colors, any of the settings that you want to show, or you can add titles. And once you're comfortable with that, with the EpiViz panel itself, you can click Save, and that would permanently save the profile to NeMO. You'll still have edit permissions, so you can still go back and edit the profile, the epigenetic data that you created, or add new tracks. As long as you don't, well, if you set it to private, only you can see them. If you set it to public, anyone on the NeMO platform can continue to use them. I think with that, I'll give it back to Joel, who's going to talk to you about how to use these profiles and create a panel and create a, well, how to use these panels and create a profile. Back to you, Joel.
Okay, great. Nice. So I'm gonna share. And so back on the home page is where you can pick the profiles that are already made. So on the left side over here under Explore Gene Expression is where you can pick pre-made profiles. But if you want to create your own profile, you need to make sure you're logged in. And then you can launch the Dataset Explorer over on the right side. So in that, once you're making a profile, it's pretty easy. You'll just click on Profile Management, and then you see the list of all the profiles that are either yours or are highlighted, so ones that we make as a curation team. But to make a new one, all you have to do is hit the new profile, and I'll just name that one as Workshop. And then you can go and use the Dataset Explorer to search for any particular profiles you want to add. So I'll just go public data sets, and we'll add one of those EpiViz panel to that. So we'll take the one that Jerem had made a couple days ago, and to add it to the profile, you can just click Add Dataset to Profile, little plus, and it'll tell you that it's added. And say, for example, we wanted to add a single cell profile to that as well. You can click on single cell, and we'll just add the most recent one that was updated to it. And now it's added to the profile. If you want to see what is in your profile, you can click on the little eye or on the right. And then you can see those two profiles that you've added. You can also change how they're laid out. So if you want to lay them out in a different way, you can grab them, move the windows around, put them in different orders. And then once you saved it, whatever arrangement you like, you can go back. And now, instead of searching for one of the highlighted profiles, you can make that search for that profile you made. I'm not sure what species the second file was, so it may not both show up. And if a gene doesn't show up in a particular profile, it'll just say gene not found. But in this case, then you see both the single cell data that was on top of that, as well as the EpiViz track down the bottom. In addition to EpiViz, there are a couple other tools that you can use for making comparisons. There's a comparison tool for bulk RNA-seq, but for today, we'll take a look at the single cell tool, single cell workbench. In this, you can pick any of the data sets that you'd want. In this case, we're going to pull up an example just for the workshop. There we go. And in this case, this data set is a bit subsampled just for the sake of running a little faster. But in this one, there's only 94 genes. But you can run it on any data set that's on the NeMOArchive platform, just running it on a smaller set for the sake of time. And this replicates a Seurat pipeline. So starting from cell counts, you can go through each step. You can select initial filtering parameters. In this case, I'm not going to filter anything because it's already a pretty small data set. And then as you go through each step of the pipeline, you'll usually get a summary graph. And then to move to the next step, you can click the toggle over on the left side for the next one. For filtering by content, and on the within each box, it gives a description of what that particular step is doing. Usually some recommendations for some initial parameters are set up already to run. And then you hit over on the right side, you can see the steps as they're running and what steps you have run so far. So in this case, it's showing the number of genes and the different counts. In the case of this particular data set, these two graphs here, which are normally the percent mitochondrial content, but in this case, the mitochondrial content has already been filtered out, so they just show up as 0. And then you can see a plot of genes and counts. And if you want to zoom in or look at any of these figures, you can click on them and they'll show up in a window, and you can download the images as well. So we're going to save this particular set of genes to move on. And then the next step is to identify genes that we want to take further into analysis. You can do that with different conventions, so either following Seurat or Cell Ranger. And the little tooltips give some more information on each of those steps. In this case, I'm just going to say use all the genes in the data set, so 94. You can either specify a number here or specify genes to add based on some of their characteristics. So either their minimum values, their dispersions, or any of these options here. But by just putting a number in here, saying we're going to look at all the 94 genes that were in this particular data set. And then it'll give you a couple plots and suggest some genes that may be highly variable within your data set. And I'll give you some plots to look at, but we'll save these and move on to the next step, which is our first one of looking at a PCA plot. So looking at some dimensionality reduction. If you have a particular gene of interest, you can put it in here to colorize the plot by that gene. So we'll stick with using GAD2. And if it's not in the plot in this particular data set, it'll show up as empty. But when you pick a gene that's in there, it'll highlight that particular gene. And on the right, it'll give you a plot of how much variance is coming on each of your axes for your principal coordinate analysis. And so based on this, the first three are providing a lot of variance. So we'll just stick with 1 or 2 for plotting our top genes. And you can add more coordinates as well to see more plots. But these are just showing how some of these different important gene or highly variable genes differ across different coordinates. And then we're going to head to the next step, which is either the t-SNE or UMAP. And we'll leave it empty because we don't have a particular gene of interest at the moment. But you can select some other arguments for your t-SNE. So in this case, the number of neighbors. And this will depend on how dense your data set is as well, but we'll just go with 4 for now. And then the number of dimensions, so the principal components, it looks like our first 3 components kind of separate out from the rest. So we'll just pick to use 3 for that. And you can change all of these settings as you go through to better refine your analysis as well. We'll run through that. That'll take a second to compute. And then now you have your t-SNE plot here to take a look at. And just out of curiosity, we'll take one of these suggested genes from further back and take a look at how it looks on our t-SNE plot. So we just drop that in the genes to colorize. And here you can see this particular gene is showing up just in that area of the t-SNE plot and not so much in other areas. We'll head on to the clustering step where you can pick what resolution you would like to cluster at. We'll just leave it as default for now. And now it's showing you the number of different clusters through your data set there. So in this case, there's 11 clusters that it identified. If you were to change the resolution, either up or down, you would get more or fewer clusters. But now we're interested in what particular genes are driving each of these clusters. And we do that on the next one step of the tool for find marker genes. And you can say we're interested in the top 5 genes for now. And you can see how they differ between the different clusters. So cluster 0 versus the rest or the other plots. And you can also pick down here, you can highlight particular genes you may be interested in. So in this case, if we were interested in these particular genes and how they differ between clusters, you can just either click for select them, or if you have genes that you're interested in, you can type them into the bar here and then hit visualize to see the expression of those genes in particular. And so you get 2 plots on showing your domain clustering of those different genes, and then the expression across different clusters as well. And then the kind of last step of the single cell workbench is to compare genes between groups. So in this case, we'll just stick with our top 5. And so if you're interested in how these differ between clusters, you can pick clusters of reference. So we'll go with we'll compare cluster 0 versus cluster 1, and you can pick what statistical test you'd like to run, and as well as the method, and run a comparison on those as well. And you can see the plots of how they differ between groups. And if you want to see the results of the, in this case, a t-test, but there are other options, you click on the show table, and it'll show you the log fold change of the different genes, as well as their p-values and adjusted p-values. So over on the left side again, right under the database name, you'll see one that says stored analysis. In this particular case, it's unlabeled because we just started it now, but you can either save your analysis if you wanted to come back to it for later, or another option is you can look at analysis as how they upload it. So in this case, clicking on the primary analysis goes through and takes the analysis that the original data set uploaders kind of tied with their samples. So before putting it up onto NeMOAnalytics, the authors of this data set analyzed it in a particular way, which may be the way that corresponds with their manuscript, or another option that they were interested in there. And if you wanted to take their data set out of that, you can go under stored analysis and pick out their primary analysis or any other data sets that are different analysis that are either saved by you or shared to you as well. So in that case, you skip all the first few steps and it brings you up to the marker gene step. But now instead of the filtering that you've applied, it uses all the filtering parameters that the authors put in to that data set. And for each of these tools, you can go and add visualizations back into a profile as well. And some other tools on our homepage is the comparison tool, which where you can pick data sets and then compare the expression of between different groups. So in this case, comparing between particular cell types. So I'll pair between motor cortex and that's there. And then you can plot gene expressions of the different groups. And you get a plot where on one side you have higher expressed in one group and the others, and then on the other side, higher expressed in the other group. You can also run statistical tests on those as well, or colorize genes by different groups. And to pick out genes here, you can either select them by highlighting, and then you can either download that gene list or add it to what's called a gene cart. So the gene carts, you can kind of think of it as a collection of genes that you can move around between data sets. So maybe if you identify 5 or 6 genes from one data set, and you want to go to a second data set and compare those 5 genes again, by adding it to a cart, you can just kind of move that data set or the selection of genes around and easily compare it between different data sets without having to go in and type out the gene name for each of those groups. And so with that, I'd like to leave a couple minutes for questions, or if anybody has other things they want to interest in, but also on the website, it has tutorials for learning how to do all the different steps of it, as well as video of the same things. So under each group, there's usually a slideshow, a walkthrough video, and as always, if you have questions or need help with trying to upload your own data, trying to find something, we do have a help desk associated with the NeMOAnalytics, and we're happy to help with any upload issues or anything like that. But you can reach us by the contact tab here. So just your name and things and a description of what you would like help with. And I think I saw a chat pop up. So one of the questions from the chat, or the question in the chat is, can you compare gene changes under different conditions rather than different clusters? So in the comparison tool is where you can have between different particular conditions. So by selecting conditions in that, maybe not donor name, but region, and you could run comparisons between different genes. So in this case, the cortex. And well, if you have a particular gene that you're interested in, you can colorize it down here to highlight it in that particular data set. But the single cell workbench deals mostly with the clusters, where the comparison tool is comparing it based on the metadata that's associated with a group.